Thank you so much. You're welcome, everyone. This is the official opening of the Noblekinmat fourth cohort bioinformatics training for beginners, intermediates, and experts. So, if you can hear me, you can unmute yourself and kindly notify so that I can know. Okay, or send a private message so that I can check myself. But I think I'm good to go since the beginning of the presentation. I have no complaints. I've heard of no complaints whatsoever. So thank you so much for being a part. My name is Akintola Ashraf. I am a PhD candidate and student at the Department of Biomedical Convergent Science and Technology, Kyungpook National University, Daegu, South Korea. This is officially the fourth cohort training that we'll be having at the Noblekinmat Limited. To serve as a little bit of introduction, Noblekinmat Limited is an impact development company. And what that means is that we identify the potential whereby we can better ourselves as researchers, as academic, as an individual, as a student. So in order to develop ourselves in our skills and also in our financial aspects too, because we are not just about learning and learning, we are also about creating an impact that will lead to prosperity and that would also be sustainable for us to sustain ourselves and to make our countries be proud of us. So you're welcome to this fourth cohort. And as I've said earlier from the introduction of the class, so there are different aspects that we'll be treating in this cohort. So we'll be traversing not just in bioinformatics, we'll also be extending our tentacles to other areas. On the day one, we'll be having, that is today, we'll be having three presenters who are well known, who have done so much in the aspects they are going to be talking about. So we have Mr. Akman Friday from the University of Abuja, Nigeria. We have Dr. Aisha Awaji from the University of Tabuk, Saudi Arabia. And we have Mr. Adebayo Kwabumi from Taipei Medical University in Taiwan. So today, our session will be based on big data, the central dogma, and the sequence alignment methodologies and approach. Two of the presentations will be in the form of lecture, and we can have a little bit of interactions after each session. And then the last presentation will be an hands-on session. So if you have your computer nearby, you can get ready to have the hands-on session. So for 1 hour, we'll be having for 1 hour or a little bit above 1 hour, because this session is supposed to start, it's between 9 a.m., that is the West African time, or the GMT +1 to 2 p.m. So we'll be having each session 1 hour, and then the third session, which is the hands-on, you can extend a little bit. And then in between the sessions, we'll be having a 15-minute break so as to give us enough time to relax and then let what we've learned from the session settle down. So the second day, we'll be having homology modeling and introduction to molecular docking. So if you are new to that aspect too, our trainer is well equipped to take us through the process. So also on the third day, we'll be having another session on introduction to Eugene, introduction to next generation sequencing, quality control, gene mapping file, and also introduction to genetics. While on the fourth day, we'll be having the ensemble training, and this will be carried out by Dr. Benjamin Moore and Dr. Alina Mushtaq of the European Bioinformatics Institute, Cambridge, United Kingdom. So they will be taking us through the introduction to the ensemble genome browser, and also this ensemble genome browser. We had the discussion, we have the training in our last webinar sessions. If you need the recording because the session is an 8-hour long session and it's free. So if you need the recording to that sessions, please reach out to me on, I would provide the link to that in our group after the presentation. If you need, you can message me and then I will share it again. So then we'll have a recap of that, and then we'll be taking through the new topics under the ensemble genome browser. So, and on the fifth day, we'll be talking about the career prospects, winning grants and scholarship, and this will be handled by a world-renowned researcher and publisher, and who has won different scholarship and awards. So Mr. Abdullah Aborodi of the Mississippi State University in the United States. And on the sixth day, We'll be talking about Cytoscape and various plugins for gene interaction analysis, various functional analysis, and introduction to 16S RNA microbial analysis. While on the last day, which will be next week Monday, because we are going to have a break on Sunday. On Monday, which will be the last day, we'll be talking about machine learning in bioinformatics and cancer diagnosis, and we'll be taking through image analysis with attraction. So please, we want every one of us to participate all through the program. There will be a certificate to the attendees of the program after the class, although it is optional. So we are also going to provide the recording of all the sessions to every one of us that attended. So this is also optional. So if you need the recording, so you have to it will be provided to you. So thank you so much. I've taken 6 minutes of our time. So I would like to call on the first presenter, who is going to take us on the topic big data, becoming big in the big data driven world. So this topic was it came about with the fact that we are in a world in which we are dealing with a lot of big data. So, but it has come to a point in which even bioinformatics also deals with big data. How do you survive in this area of big data? How do you manage yourself, and how do you improve yourself in becoming one of the experts or one of the beneficiaries of this upcoming, or it's not even upcoming. So in this popular aspect of our worldly endeavor. So to take us through is a very, very good presenter, and has also traversed that path of accessing opportunities, accessing the potentials of bioinformatics. So, and he's no other person than Mr. Akpan Friday. So you can, Mr. Akpan, you can come in and share your screen so that the presentation can begin. So if we have any question before, as Mr. Akpan shares the screen, so we can send it to me on a private chat, and then I'll be able to read it out to every one of us and then respond to the questions accordingly. Thank you so much for being a part. And okay. We can share the screen. As we await the screen to be shared and the presenter to come in, so okay. Okay. You can unmute yourself. Okay, I'm coming. Let's see. Okay. Now, I think you can speak now.
Thank you, Afshal. Can you hear me?
Yes, I can hear you loud and clear.
All right. Thank you. Can you see my screen?
Yes, you can speak.
So thank you, everyone, for joining in. My name is Friday Avan, as Ashraf has said, and thank you very much for joining into this Noblekinmat Bioinformatics training. And I'm so glad to be here. My name is Friday again, and I'm a student at the University of Abuja, Department of Microbiology, and I'm very much interested in bioinformatics and precision medicine. And today we'll be talking about, I want to really give you an overview of why this course will be very useful to you. And that's why I'm talking about big data and becoming big in this big data driven world. The world is full of data now, and there are a lot of data generated every day. and I hope that this course will actually give you an insight on why you should become big in this big data-driven world. So I want to really start by saying that I have taken a lot of courses in school and at the University of Abuja, and only a few were very, very memorable to me and quite useful for me in my later studies. And I hope that this course, and I know that this course will be certainly one of those type of courses that's kind of memorable and very useful to you in your later studies, whether as a researcher or as a student or a scientist. So without further ado, let's move on to what we have today. So I kind of want to give a big overview of what exactly is bioinformatics, and we're going to move into the big data-driven world. So kind of saying, why should you learn bioinformatics? It's kind of the data generated we have these days. And I want to give you an insight to some of the applications of bioinformatics and how this training will be very useful to you, and some skill areas of bioinformatics, how you can learn bioinformatics, this training is going to do for you, and the opportunities open to undergraduates. I really might not need to cover that, but we'll see how far we go. So what exactly is bioinformatics? I'm sure you're already enrolling for this webinar series, and I'm sure you already have an idea, but we kind of want to remind ourselves exactly what bioinformatics is. And if you probably have come across different definitions, sometimes you even get it confused with computational biology, which is kind of really almost the same thing, as would say. But then let's look at what bioinformatics is. So Wikipedia actually takes bioinformatics as an interdisciplinary field that develops methods and software for understanding biological data. By interdisciplinary, we can look at this sort of Venn diagram, which is kind of overlapping, where we have bioinformatics in the middle, and we feel like it consists of different fields, both computer science, statistics, biology, biochemistry, mathematics, chemistry, even engineering. All these fields coming together gives you, bioinformatics. But the two main fields being both computer science and biology, with statistics, a bit of statistics. So bioinformatics is actually that field we really want to understand data. We want to understand information, biological information. And the National Human Genome Institute actually defines bioinformatics as a subdiscipline of biology and computer science concerned with the acquisition. How do you acquire this data, store data, analyze data, and disseminate this data? So all these things, we can see they keep on talking about data. What is this data all about? Biological data. We're going to see that in a bit. So next slide, we have the big data issue. What's this big data issue in biology that we are talking about? We've been hearing that the data explosion, a lot of data generated. What's this data? Well, this data can be anything. You could have DNA sequences, protein sequences, sometimes even the way you live, your X-ray, the way you eat, all these things combined together are big data, not just limited to DNA sequences or nucleotide sequences or protein sequences. All these things combined together are, what we call big data generated every day. And what we say is a big data, and how do we get this big data from? Well, sequencing technologies since 1953, when the DNA was discovered, we've had constant improvements to DNA sequencing DNA, and the DNA consists of millions of letters, ATG and Cs that we have. Then we look at it and we can sequence a whole genome, a human genome. We can sequence organisms, their mitochondria, and we generate this whole lot of data every day. Bench work on DNA, proteins, and metabolites have aided the production of biological data available on any topic, including genomics, ecology, diversity, biodiversity, epidemiology, and etc. So technologies that generate these biological data sets are now kind of routine part of our biological research. We have to do them every time. People have to sequence DNA to find what this part of the gene does, what this part of the DNA sequence does. So, it's kind of part of biological research now. And people are, these technologies are improving every day. We're going to see how that works. And now you can see in this graph here where you have data explosion. And it says that the amount of genetic sequencing data stored at the European Bioinformatics Institute takes less than a year to double its size. So, just imagine you have all these data. You can even imagine the data on your phone, and you can say, imagine the data on your phone doubling its size. But now you have this massive amount of data doubling its size at the European Bioinformatics Institute in less than a year. There's a lot of data explosion. Data sets are just so massive right now. So I want to use the human genome as a very good example. We can see here in 2001 on this slide. In 2001, it cost over $100 million. In 2001, $100 million to sequence a whole human genome. And I'm sure you have an understanding of the human genome. The human genome consists of over 3 billion base pairs. So you can just imagine A's and G's all filled up, over 3 billion of them. You can imagine. And it cost over $100 million in 2001 to sequence a whole human genome. Now, at that time, people obviously wouldn't want to sequence because, of course, that's too expensive. That's too expensive for an individual to say, I want my whole genome, sequenced and all that. But looking down, we can see a kind of decrease, a kind of small decrease up to like 2007, where it was kind of following Moore's law. And if we can see that little decrease, it was because of kind of increase in improve in sequencing technologies. That was why we kind of saw that decrease in amount up to a kind of $10,000 million up to 2007, where we kind of had high throughput sequencing, where you don't just sequence one stretch of DNA, but you sequence multiple stretches of DNA at a time. And you can see that, of course, it didn't even follow Moore's law again. We can see a massive decrease into the amount of which we use in sequencing a human genome. And now in 2021, what do we have? In 2021, sequencing your whole genome costs less than $1,000. So if it costs less than $1,000, people are actually doing it now. We have these data sets, and then you can go and say you want genome sequenced. And if we can see you want your whole genome sequence, we kind of have a lot of data now on this, cost of human genome, 3 billion base pairs. But it takes because of the improve in sequencing technologies, we can now have a human genome sequence for less than $1,000. And even when we had it for over $100 million, it wasn't really sure of what people would learn from it, what scientists would really learn from it. But now we can see that with the improving sequencing technologies and computational tools, we're going to learn a lot from it, and then people really want their genome sequenced. So I hope you all got that. Okay.
Yeah.
So yeah, you can all hear me, right?
Yes, we can hear you.
Yes, cool. So see, these large data we are now generating, we are generating large data sets every day, every minute, every second. Every day, every minute, every second. And we can now see people are using for personalized medicine, for a lot of research, and the world is becoming really, really, really diversified. So another thing I want to show you is an article I came across recently just on the 23rd of July, this 2022, and we saw that UK, the United Kingdom, is exploring whole genome sequencing for newborn babies. Whole genome sequencing, where a human genome, as we said before, consists of over 3 billion base pairs. 3 billion base pairs. And you can imagine the UK is, kind of exploring sequencing for newborn babies. They want to sequence their whole genomes, where you can actually say, okay, this part of the genome can code for this part of this, and try to identify genetic diseases and susceptibility to drugs and all that. So with kind of exploring whole genome sequencing for newborn babies, we can now see that there's going to be, large, more large data sets. You can imagine where they kind of store all the data sets. If all the babies being born and they are sequencing over 3 billion base pairs for one baby, you can imagine. So, still on this big data issue, I have this quote from Sidney Berdner, who is a 2002 Nobel Prize winner, and he's a biologist who has done experiments all his years. He actually identified all the cells in C. elegans worm, and he has done experiments all his years, and he's been doing things. And he made this quote during an interview, and he said, "We presently have huge amounts of data that are largely unassimilated. I have discovered that while there are rewards for collecting data and distributing data, there is little, if any, support for organizing it. It is what I call low input, high throughput, and no output science." Now, this kind of emphasizes the need for bioinformatics. Why we need bioinformatics. Now, it's just saying that, we're kind of having a lot of data these days. That was in 2002. He said that we're having lots of data, but we have little knowledge on how to really organize this data. And it's really kind of a big problem, a very big problem. If you have your data and you cannot analyze it, you cannot actually make meaning from this data. It's kind of useless if you cannot actually make meaning from your data. Meanwhile, low input, high throughput, and no output science kind of mean that, scientists actually do not really know what went. They just kind of, we have a sample, let's sequence it. And then, they don't have any idea on how to, use computational tools to analyze this data. And then, of course, no output science. We really don't have anything. We just kind of have a bunch of data without no meaning. So it's very, very much important for us to understand computational tools as a biologist, someone who's been met as a student, as a researcher. It's one of the core competencies of 21st century, personalized medicine, and, a skill required by almost all life scientists. So again, still on this big data issue, I got this paper, I came across this paper which says all biology is computational biology. Now, you want to ask yourself, as an experimental biologist, as a student, as someone who's in the life science, as a researcher, as a scientist, can I afford not to learn computational biology? A very big question. Because experimental biologists take years and years and years of, developing new methods to experiment for their data. But then, if you can take new methods and to learn on how to carry out experiments, but then you're going to spend, a lot of time sitting down in front of your screen, thinking of how you kind of analyze that, those methods you've developed and those data you've developed. So computational biology is very, very, very much important. And it's very important to understand that without the advent of modern computer technology, we will not be able to analyze the DNA sequences that we have. And understand that it is those that can be applied to individual patients or individual organisms. We can check for, DNA sequences, protein sequences, nucleotide sequences, and say, okay, this is this region that codes for this. And identify regions that codes for eye color, for a particular trait, any different kind of traits. So I think, this excitement in personalized medicine, in, big large data sets, it's now due to the new tools that we have to understand variability and to exploit, these large data sets that we have. So one of the signs of I think that is taking advantage through this development. One in the science of DNA, and the other one in the science of managing large data sets. So large data sets are not just DNA. Large data sets are not just DNA, but other kinds of data sets that I can mention before. You can imagine that you could just like you can surf Google for any information, you can also surf your DNA for any kind of information, but you have to do this with computer tools. And big data is another way of looking at very, very large data sets and trying to understand what it is about an individual person, individual organism, what makes them different. What really makes them different? So by looking at all these patterns of DNA, proteins, we'll be able to measure, look at disease susceptibility, look at DNA patterns, and kind of trying to understand what it is that makes someone or an organism really, really, really different. So another thing you must understand is that bioinformatics cuts across all fields of the life science. So what are I heard people kind of ask me what exactly should I learn in bioinformatics? Why should I learn this? Bioinformatics cuts across all fields. So what are you studying computer science? You really want to in this age you really want to kind of dive into bioinformatics and computational biology. But people say, I'm interested in antimicrobial resistance. Is there bioinformatics for that? Yes, there is. I'm interested in cancer research. Is there bioinformatics for that? Yes, there is. I'm interested in personalized medicine. Is there bioinformatics for that? Yes, there is. For vaccinology, for dermatology, for crop improvement, for forensic science, for immunologists, for drug discovery, all these things, bioinformatics finds application in all these fields of life science. And that's what this course is all about, to give you an insight to see how can you apply bioinformatics in your own area of research, in your own area of interest. How do I apply bioinformatics in these areas? So, it's quite very, very much useful, and one of the core competencies of today's, 21st century science. People want to see how, they can use computer tools and softwares to actually analyze these big and large data that we are seeing everywhere. So, I want to go into why you should learn bioinformatics. Why should you learn bioinformatics? And I actually prepared this slide for mostly undergraduates, but it's quite applicable to everyone who researches, for students, for industry experts, and anybody who is actually interested in going to the field of bioinformatics. These days, we actually see applications thrown out. A lot of applications thrown out, and then people are asking, you kind of go through the eligibility criteria, and you see whether you have experience using R and Python, and you kind of say, well, I'm a biologist. What does this really have to do with me? Why do I have to learn programming languages, or why do I have to have an idea on Python and R? It's kind of emphasizing the need. Right now, people are actually knowing that, you need to have a bit of computational knowledge, how to analyze data. How do you run simple algorithms? How do you use simple software tools? How do you run BLAST? How do you compare similarities between organisms and sequences? So the frequent questions lab and industry are asking is if you have experience using R, Python, or Perl, if you have basic computational skills, do you have experience handling data? You see some saying experience using Bash is a plus. Are you familiar with the command line? Do you know how to just analyze data do you know how to do phylogenetics, proteomics, metagenomics? These are questions that are being asked when people are hiring and trying out applications. And I have this quote here from Lilith Nessian, who at a TEDx, where she said bioinformatics is one of the most needed and hard to find competencies of the 21st century. So we know that bioinformaticians are still very few and low, especially in low and middle income countries like in Africa. And this is one of the purposes of this webinar series. We are trying to upskill 1 million bioinformaticians, 10 million bioinformaticians, as many as we can, to actually, upskill them, because bioinformatics is very, very much important in this 21st century. So another reason why you should learn bioinformatics is if you're considering going into technology, or you love the computers and you still don't want to go away with the biology. Bioinformatics, as we said, is quite interdisciplinary. It brings together both computer science and biology and every other field. It brings them together. And if you really want to go into, if you are considering tech and also biology and statistics, it brings all that together, and you'll find a very lovely time engaging in bioinformatics. So you can have both all these fields. If you are kind of the people that love to cut across all fields, they want to know a bit of statistics, they want to know a bit of biology, a bit of computer science, a bit of engineering. You can get all that with bioinformatics. So another reason why you should join bioinformatics is you should join global rules of innovation in personalized medicine and genetic engineering and related biotech development. You can see that everyone is clamoring for personalized medicine, because this idea of treating patients individually, it's something, everyone is different. It's something everyone is looking into, and people are welcoming, and bioinformatics is at the forefront of this, and even in every kind of research. So if you kind of want to know some little applications of bioinformatics uses computer tools to track and analyze patterns of disease outbreaks like COVID-19 around the world. Bioinformatics was very much useful when the COVID-19 outbreak came out, and people kind of use bioinformatics to actually analyze these patterns of DNA sequences around the world. So what are these core skill areas in bioinformatics? You want to check out computational skills, interpretation and scientific skills, statistical and mathematical skills, and communication skills. Computational skills is just talking about how you use your system. Interpretation and scientific skills is really talking about your own intuition. How do you analyze these biological data? Just as we said, you can't be a bioinformatician without understanding the biology behind it. So whether you are into computer science and thinking of how you can dive into bioinformatics, or you are a biologist, experimental biologist, a researcher, a student, you have to know the underlying biology behind everything. Even if you are using computational tools and software tools to analyze it, you have to have ideas of this biology. You cannot do away without the statistical and mathematical skills, where you talk about kind of testing, hypothesis testing, ANOVA, and all kinds of tests you run with bioinformatics. And communication skills really talks about visualization. How do I interpret and tell people about my data and showcase my data outside? So just as I said, we kind of just want to skim about each of them one after the other, but I'm sure they're going to give you really good insights to a lot of beginner areas for bioinformatics, and kind of all encompassing whether you're a student or a researcher, those key skill areas you have to learn about bioinformatics. So if you want to go into the computational skills, where you kind of have programming languages, you learn the Linux command line, which is kind of the gold standard kind of the operating system, which is more widely used in bioinformatics. You want to learn about workflows for bioinformatics, databases, algorithms, and data structures, and all other computational skills you need to have in bioinformatics. So, just like I said, interpretation and scientific skills, you have to have that biology interpretation, that intuition. How do I understand biological data? A computer scientist might be able to do the whole programming and be able to use software tools, but you have to really understand the underlying biology behind it, biological significance and applications. So you have to really understand, DNA. You have to really understand proteins, what are the central dogma, and you really understand what the kind of data you're dealing with, biological data you're dealing with. So it's something this training is going to do for you. I think we are starting with dogma of molecular biology, and you're going to learn about the DNA sequences and proteins and translation, transcription, all these processes. You're going to learn it in this training. Believe me, it's going to be really worth it. So just like I said, statistical and mathematical skills, where you talk about testing, hypothesis testing, multiple testing, machine learning, and even probability. What's the probability? And even when you run blasts, all scores, and all these things, all are all parts of statistical knowledge you need to understand. As we said, communication skills too, where you have data visualization, reporting of data, explaining methods in less technical terms, because as a bioinformatician, sometimes you might be tasked with, explaining your data, the data you've gotten and you've interpreted and analyzed, explaining it to people who are not really, into the field of bioinformatics or don't really have a knowledge about it. So you want to explain it in less technical terms, and these are the kind of things you really gain engaging in this training. So where do I learn bioinformatics? I think you're in the right place already. This is bioinformatician. This is the right place to learn bioinformatics. Come during our trainings. Throughout these trainings, you're going to learn a lot. And I believe me, your certificate, is very important, because you're going to learn a lot throughout this training. This is the right place. How do I start? Start by taking this training. Do not miss every part of this training. What should I learn in bioinformatics? This is the right place. Don't worry, you'll be guided. If you need mentorship, you could always ask questions. These are the whole things you would learn during this training, and you could always measure your progress. So, okay, I'm just gonna move up to all this. So I kind of have some free resources, bioinformatics courses for all these. If you, I think they are all found on this Telegram channel. If you want to get some more resources you can use in learning, you can find them on this Noblekinmat Bioinformatics channel. And also, sorry, someone said they've lost the audio. Can you hear me? Sorry, can you hear me? Sorry, I don't know. Can you hear me? Ashraf, can you hear me?
Yes, I can hear you loud and clear.
Okay, someone sent me a private message. We've lost the audio, so I kind of had to pause.
It's
okay.
The problem of the network from their side. The audio has not been faulty all through.
Okay, so you throughout this presentation, you've actually understood big data, the problem of big data, and the need for you to actually take such kind of training, because it's one of the core competencies of the 21st century. Engaging in this training would give you much insights to what bioinformatics is. You would understand bioinformatics and be able to, you will never be afraid of big data. Again, as a researcher, you will never be afraid of big data. So please stick to training, and I still have a lot of opportunities down here in the slide, which I'm not going to talk about now. You could, if you need the slide, I could send it out. You can find it on the group here. And in conclusion, bioinformatics is really a growing field, and bioinformaticians are still very few in low and middle income countries like Nigeria and in the whole of Africa. Bioinformatics skills the key tool in modern science. So it is therefore required of undergraduates, researchers, scientists, and everyone to arm themselves with bioinformatics skills and join the global race of advancement in science in the data-driven world. So thank you very much for all joining in for this presentation, and stick through the whole webinar series. And I want to say a big thank you to Ashraf Akintola for giving me this opportunity to give this presentation. Thank you very much.
Thank you so much. This is really, really insightful, and I so much appreciate your resourcefulness. I will take you from the last part you finished presenting. uh, presently the internet is swarmed with free resources that once you get to sit down and study everything, you are becoming a seasoned bioinformatician yourself. That is one good thing about this aspect of learning. So you have every resources at your disposal, and you can go into any field as much as possible. So these guidance are present free of charge. Of course, there are paid courses. There are also many paid courses, but there are also a lot of free courses like this that we are having here. A lot of people got different opportunities, different exposures, different proficiency level from attending our webinars. We've had experience of people going ahead to win awards from this webinars like this. So and I know that everyone that is new to this our program would also benefit immensely from our sittings. So if we have any questions, so I think to ease the communication and to make it easier for us to reach out to questions, I am going to share a group whereby we can send in our questions after today's session, or I can send it during the break, because we'll be having a 15-minute break before the next presenter comes in. So so that we can send in our questions and then we can interact more. Although we have WhatsApp groups, and we have the WhatsApp groups are filled already, so that's why it is better if we have one Telegram group that can take as many people as possible, and not just the channel. The channel is just to reduce the traffic of messages in and out, so that we can have the goal can be very to trace our messages. So I'll create a Telegram group. I will share the link on the channel so that we can drop in our questions, so that you won't be sending it to on the personal message, so that everybody can also benefit from your questions and also see the answers to them. So thank you so much, Mr. Friday Afan, for the wonderful presentation. So bioinformatics is a very wide field that everyone can take. Anyone, you're in mathematics, you're in statistics, computer science. We've had people that are in law attending our webinars. We've had people in business attending our webinars. So as long as you know what you want to achieve from this science, it's very easy. You can learn it. It's just a matter of time and commitment and internet access, a very good computer, and you're good to go. So that's about bioinformatics for you and the big data. Thank you so much. If you have any question, you can kindly send it to me on the private chat so that we can read it out, and then we can continue in our presentation. So we'll be having, if there is no question, we'll be having a 10-minute break, because we've eaten deep into our time. So we're having a 10-minute break, and then by 11:05, we'll be starting the next presentation. Thank you so much. Okay, we have some questions here. Mr. Akman, don't go yet. So the question says that what is the characteristics of a big data? Can you help us answer that?
Okay, you said?
What is the characteristics of a big data?
What's the characteristics of big data?
Yes.
Okay, so I mentioned that I think I still remember mentioning that that large data sets are not just DNA, but all kinds of data sets, and it could be proteins, it could be nucleotides, and when you surf at Google, what your X-ray looks like, even what your shopping habits looks like, and taken together, that is defining the new science of all what we call big data. All those things are defining the science of big data. Now, I want to tell you, exactly if you're talking of data, for example, I mentioned the human genome project, where you kind of sequence a human genome, and a human genome is over 3 billion base pairs. Now, informatics helps us to do something that something that is humanly impossible to do. Now, you know that DNA sequences are A, T, G, and C's. Now, if you have over eight, okay, let's take for example, you have over 3 billions of those letters written down. How do you sift through this enormous amount of data in a way that it would be inhumanly impossible to do? So that's where bioinformatics comes to play. This is a whole large data. Even if you have, a mixture of A, T, and G's all fixed together, and you have or protein sequences or nucleotide sequences all fixed together and scrambled, and you're asked to count the number of T's, you can imagine how long it will take you to do. Or count the number of T's where you want to find 80% to take you a whole lot of. So all these things are, like we mentioned, are just what's coming up, and a lot of improved sequencing technologies. If you go to sites like the NCBR, you kind of find out that there are a lot of sequences that have been sequenced and splitted, or I kind of saw recently, days back, that AlphaFold have solved almost all known proteins, found structure of all known proteins. That's AlphaFold and DeepMind. They solved the structure of almost all known proteins. You can imagine we have the structure of all known proteins right now. That's kind of data too. So there's a lot of data being generated, and this is what is defining the science of the data.
Okay, thank you so much. To also add to that, we all remember the type of the first hard disk or memory card that was first created. It was put into a very big trailer, so just I think 1 megabyte or so. Now we are having 500 and more than 1 terabyte on the phone. So that means the data that are required, that are being collected, we remember the era of a Sajem, whereby if you have 10 MB, you can browse. So that means there is an influx and explosion of data presently in our world. So how do we analyze this data? So that is what this big data also explains, in addition to what Mr. Akron Friday explained. So there's another question here: is that is there an overlap in informatics and data analysis?
Yeah, okay. In my own view, data analysis, might not really be specific to biological data. So, bioinformatics is actually analyzing biological data itself. So data analysis can be, all types of data, but you kind of have other types of data that can be generated anywhere. Even in schools, attendance is a kind of data. Your own attendance anywhere else is a kind of data. People's shopping habits is kind of data. Even patients, records are types of data. But organizing, biological data specifically, is what we have bioinformatics. If we want to talk of overlap, we might talk of bioinformatics and computational biology. But bioinformatics and data analysis were quite really different. Data analysis might just be analyzing any type of data, but which really is not biological data. But bioinformatics is analyzing biological data.
Yeah, thank you so much. There are, just like you explained also, in addition to that, because bioinformatics also deals with a lot of big data. So having another skill set of data science or data analysis skills is an addition to you in order to infer the meaning from data, from biological data. So if you have that skill set, just like a resource person said in the presentation, so he also mentioned the data analysis and other stuff during among these specializations that you can also venture into. So, for example, in data science, there is this topic of cloud computing, statistical analysis, data visualization. So when you are doing biological bioinformatics analysis, you'll be doing a lot of visualizations too. So not everything will be the code running and you'll be seeing the result. No, it should also bring you down to data visualization. So that is when you now have that combined skills of bioinformatics and data science, you are a hot cake. I don't know what other term uh, to say it. I don't know what other time to clarify your resourcefulness. So because you are dealing with a lot of data and you know what the bioinformatics aspect is and you know the data analysis aspect is. So there is a lot of it's interwoven. So as a bioinformatics, you can deal with the biological aspect of data analysis. So it doesn't necessarily mean that you know all the aspect of data science or data part. So that's just the to buttress the point of our speaker. So other questions that I've been seeing here are some of the questions that will be answered later. So like bioinformatics, how do you transition as a biochemist from into bioinformatics? So that we are going to answer that in details in after the presentation so that we can have time for recess and so that we can have time to rest before the next presentation. So thank you so much. We are supposed to resume 11:05, so we have 5 minutes more. So we have back here in 11:07 Nigerian time. We're going to back 7:07 South Korean time. So we have 5 minutes to relax and recline before the next presentation. Thank you so much. That was the first session and it was impactful as promised. So if you have any more questions, I would please hold on. I will create the channel and the group and then I will share the group link on the Telegram channel. Thank you so much. See you in the next 5 minutes. So to join the group, the link has been shared to us on the channel. So you should be able to drop your questions. Okay, thank you for being here. So I think I have to allow Dr. Aisha to speak. Dr. Aisha,
Yes.
you can share your screen now so that we can start the second session.
I think there is a message that the previous speaker is sharing the screen. So, yeah.
Mr. Akpan, can you stop sharing your screen so that let's see. Oh, 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 oh. Stop sharing. Mr. Akpan, I think there's a way to. Let me do this. Let me remove. So I think it's not around the system currently. Please hold on. Let me call him. Probably it's not around the system. Boss, please, can you help us stop sharing the screen? I think Dr. Aisha wants to share her screen. Yeah. Sorry. I would attend to. Okay, now, Dr. Aisha, you can share the screen now. I would also attend to the questions, one after the other. Sorry for the delay. Yeah, good. So thank you so much, everyone. as we are about to begin the second session, so let us inform others who are still not here present to be back here for the second session. So for the first session, we have a brief introduction into what bioinformatics is and a little bit of the scope in which you can explore in the aspect of bioinformatics. And now we are now streamlining our focus on the main aspect of bioinformatics. So which is I think my voice, can everyone hear me?
Yes, I can hear you.
Good. So so which is the going deeper into bioinformatics, which is the area of genomics and a little bit of transcriptomics and epigenetics. So if you can't hear me, I think it's probably an issue from your side. So please try to rectify that. So anyways, so our next presenter is a very, I don't know how to describe her. So she is presently, let me take it from the professional angle. So she is presently an assistant professor at the University of Tabuk, Saudi Arabia. She had a PhD from the University of Leeds, and work focused on cancer. So, and she's basically focused on the medical molecular biology with genetics. So you are on to an expert in this field and who has a vast level of experience in discussing the topic, the impact of genotype-phenotype association on health and disease. Ladies and gentlemen, let us welcome Dr. Aisha Awaji, all the way from Saudi Arabia. So over to you, Doctor. You have the floor.
Okay, thank you, Ashraf. Hello, everyone. So I am Dr. Aisha Awaji from Saudi Arabia, University of Tabuk, and I'm going to present today some of the background about the central dogma of molecular biology, which is the basic knowledge the bioinformaticians basically have to know about when they want to analyze any data related to genetic variations and genetic variation and their associated diseases. More I would say different diseases including cancer. In this session, I'm going to talk more specifically about general information in the DNA representation, so the structure of the DNA, and then I'm going to, and then we move to the central dogma overview. So central dogma of molecular biology, including the flow transformation of the genetic information, DNA to RNA to protein during transcription and translation. And we will focus, and then we are going to move to to see the effect of different types of genetic variations on the gene coding or coding sequence or non-coding sequence. Also, and then we will see the effect of these genetic or the phenotypic effects of this genetic variation on the sequence of the DNA and the production of the sequence of DNA at the RNA and protein level. Then we are going to talk a little bit only about the high throughput techniques that allow us basically to study the genotype, phenotype, including genomic, proteomics, and transcriptomics and proteomics and metabolomics. So to start with, we will have brief introduction. We know that DNA plays an important role in determining our phenotypes, our traits, and health. So our eye color, hair color, and height and weight, and all the phenotypic characteristics, and also our abilities to develop any diseases. We know that also different types of human cells contain the same genome, but also show different structure, and also a different cellular structure and different functions as well. Our cells contain about 3 billion base pair of the DNA, and all these DNA compacted by the DNA packaging in the nucleus. And also we know that during the cell division, DNA organized into 23 pairs of chromosome, in which at the end of the cell division, 2 daughter cells are produced, and each one of these cells has the exact copy number of the DNA. While outside the cell division, we know that the DNA is decondensed for greater accessibility. So we know that humans in general are genetically identical at a percentage of 99.9%. But we still see differences. We see still differences in hair color, and skin color, and in height, and weight, and for example, in progressing any diseases, and so on. So from where these, so if we are similar, about 99%, so from where these differences come? We know that human genome, so to answer that, we know that human genome consists of about 20,000 genes. Only less than 2% coding genes, so they produce a protein, while the remaining about 90% is non-coding, so they don't produce any protein. And the genetic differences, or the genetic differences among humans accounts for 0.1%, and this leads only this small percentage leads to produce like 3 million differences across the genome within the 20,000 genes. These genes are found in the protein coding genes. And these differences actually create the genetic differences that we see amongst humans or at the population level. So understanding the gene structure, so understanding the gene structure, protein function, and all the genetic and basically the nature of them, the nature and the type of the genetic variation, and the impact of these genetic variations to cause diseases would allow us to understand the disease, understand how to treat the disease. So this would eventually lead to to improve health and medical care. So moving to representation, so if we want to basically to analyze or to identify, to analyze any variation in human genome, or using DNA sequencing, for example, analysis, we have to know the representation of the DNA, so the structure of the DNA. So here I'm going to a brief description to the structure of the DNA. So DNA, we know that DNA, a nucleic acid, and consists of building blocks called nucleotide. And we know that each nucleotide, I will move to the picture to understand the structure of DNA. So we know that each nucleotide is composed of three features, which are sugar, and we know that the sugar 5-carbon sugar, and the sugar, the type of the sugar is deoxyribose sugar in the DNA. So the sugar linked to at the first carbon of the sugar linked with nitrogenous bases, which in the DNA, thymine, adenine, cytosine, and guanine, while the 5-carbon in the sugar linked with a phosphate group. Also, we know that the sugar and the nucleotides in one strand, we know that the DNA has double-stranded, and the nucleotide of one single strand linked with each other by sugar and phosphate, by phosphodiester bonds that link the sugar and the phosphate. Okay. So the sugar and phosphate, so the phosphodiester bonds links the nucleotide with each other. And also, we know that the double-stranded binds with each other by hydrogen bonds. The hydrogen bonds that leads to pair the thymine with adenine and the cytosine with guanine. We know that there is a 5-end and a 3-end, 5-end or 3-end of the DNA strand. So as we can see in the picture, the 5 ends and the 3 ends. So the 5 ends mean that the 5-carbon in the first nucleotide of the sugar in the first nucleotide has a free or is free of binding with any elements before that, and also of free phosphate actually. And the 3 ends that mean the 3rd carbon in the sugar and the last nucleotide in the strand is free. So the hydroxyl attached with the 3rd carbon in the sugar and the last nucleotide in the strand does not attach with any nucleotide after that. Okay. And we know that the 2 strands are linked with each other, as I said, with hydrogen bonds, and the strands bind to each other in opposite orientation. As we can see, so the 5 ends correspond to 3 ends in the opposite strand, and the 3 ends in one strand correspond to the 5 end in the other strands. So usually the length of the DNA is measured by of the DNA double-stranded measured by the base pair, while the single strand measured by nucleotide. So also, during writing the structure or writing the sequence of the DNA sequencing, it's important to know that writing is from 5 to 3 direction. And for the another strand that started from 3 to 5 is also the same, since this strand is also opposite is bound to the first strand in opposite orientation. So the sequence also on the second strand written from the 3 to 5 direction, as you can see in the picture. So why we are focusing on the DNA presentation? Because DNA usually has DNA sequencing, and these sequencings are important for basically for people or for researchers who study disease or genetic variation, in which can analyze the DNA sequencing and identify any genetic variation. As we can see, for example, here in the patient 1, patient 2, we can see some genetic variation with them some genetic variation in the base pair T to A for patient 1 and GC in the patient 2. So when we move to the central dogma overview, I would to make it simple. So the central dogma. So why we are studying the central dogma? Because any variations or any mutations affect the gene sequence, which is the genotype, would also affect. Is not always affect, but maybe it depends on the variation, or depends on mutation. May affect also the phenotype, which means the protein produce it. So it's important to know the DNA looks like, the RNA looks like, and the protein looks like. Okay, and what is the component, or what's the feature of the DNA of the gene? What's also what is the features of the RNA? What's the feature of the proteins? So all of these information would help you to analyze your data. For example, your genes data, your transcriptomics data, your proteomics data, and so on. In the concept of bioinformatics. So. We know that the central dogma it's also the flow of the genetic information from DNA. So it's a DNA sequence, a double-stranded DNA in which transcriptize into RNA by the transcription machinery, and this leads to produce primary RNA. So the primary RNA is a single strand that has a similar sequence to the coding gene strand, which is the 523 usually, except the letter thymine, which is replaced by uracil in the RNA. So after the primary RNA is produced, it undergoes to a specific process called RNA processing or modification, in which it's like clearing. We know that clearing or making the DNA sequence clear from any regulatory region or any regulatory sequence, including promoters and so on. And we will talk after that. We'll talk in the next slide about it. Make the sequence of the primary RNA also clear from any non-coding sequencing, and we will explain it in the next slides. In the next slide in this lecture. So, as I said, the primary RNA undergoes to RNA processing. To produce mature messenger RNA, and after that, the mature messenger RNA will include the coding or the genetic codes that the genetic code, which can be transformed to the specific amino acids to produce the polypeptide, or the polypeptides which then modified and folded and modified to produce the protein. The proteins will have specific cellular function or structure function in the cell. So basically, DNA carries the genetic information, which is the genotype, and this leads to direct the RNA production by transcription, and then RNA direct the protein production by translation. So moving to the transcription, which is the first process in the central dogma. So what is the transcription? Or transcription? It's like transcribes all or copy the DNA. This is basically so copy the DNA for gene expression. So before explaining the transcription, I would to describe the gene structure. So basically, as you can see from the DNA, this is if we imagine that there is this is only a fragment of the DNA which includes the gene to be transcriptized. this gene would have specific elements or specific features that allow the transcription to occur. So, for example, the enhancer, the promoters, the transcription factors, which attach to the promoter and enhancer to initiate the transcription, and also the gene body. So the gene body is still gene body including exons and introns, and the terminators. So basically the promoter. It's like the key of the transcription, the terminators switching off the transcription, okay? And also RNA polymerase II are essential enzymes that play, as essential enzyme play important role in transcription of the DNA sequence into RNA sequence or into primary RNA. So once the transcription, is done, the primary RNA is produced, and as you can see, now it's make visible that the primary RNA contains of exons and introns. So the primary RNA needs to be transferred to the mature RNA to allow producing the protein. So in order to do that, so the primary RNA undergoes two specific RNA processing, which is so the RNA, which is include 3 steps. These steps you can see in the mature RNA. These steps is the capping, which is attachments or link of specific chemicals at the 5 ends, and also polyadenylation, which is forming poly-A tile at the 3 ends. These play a feature role in help or in protection of mature RNA from degradation when it's transferred from the nucleus into the ribosome, because the transcription itself occurs in the nucleus. So in the nucleus, everything is protected. Once, so the RNA processing, including the capping and poly-A tile, it's like a science. It's like a science. These signs help in protection the RNA from degradation when moves to the ribosomes. And the last step, so the first step capping, the second step is poly-A tile, the third step is splicing or intron splicing, which means removing all the introns and keeping the exon. So the exons contain the coding sequence, which means these sequence will include it in the protein coding region and will produce proteins. While the introns, non-coding sequence, which means they don't produce any protein, so they have to be spliced, so removed. So introns are removed and the exons are remained. Okay. So by the end of the transcription, mature mRNA is produced and consists of the protein coding region, which is the exon. Okay. So, and then moving to the translation. So we move to the translation to the, if you can, to the mature messenger RNA. We can see, so the translation is, so the translation is producing the protein. So mature messenger RNA also has a specific structure. So what are these structures or specific features, I would say? So if we can see here, we can see that the protein coding regions, which are the exons sequence, the first the mature messenger RNA contains sequences like start codon and stop codon. And if you can see also, before the start codon, there is before the start codon, there is a 5 untranslated region. And after the stop codon, there is 3 untranslated region. These two regions, they don't involve, actually, they don't from their name, they don't translate, they are not translated, but they will, but if there is any mutation in these location, these regions, that would affect also the proteins level, the protein levels, but they don't affect the RNA, the protein sequence. So let us focus more in mature messenger RNA features, as I said, which are essential to transform this sequence to amino acids. As I said, the mature messenger RNA include the start codon and the stop codon. These codons will be the signs for starting, signs for the ribosomes, where to start translation in help with other RNA, for example, the transfer RNA that helps decode the sequences on the premature messenger, in the mature messenger RNA into amino acids. So the transfer RNA will help in decoding the coding sequences. For example, AUG is the start codon, so the amino acid is methionine. Okay, and the same for the rest of the codons, until the translation is terminated at the UAA, which is the stop codon, where the translation is terminated. And this leads to produce a chain or amino acid chain or polypeptide. This polypeptide is folded and modified to have specific role, or to have specific role in a specific function or structure in the cells. So now we will go more deep and to understand how the genotype actually, or how the genetic information, how the genetic variation affect the genotype and affect the genotype, which also show an impact or influence on the phenotype and the protein function. So type of genetic variation. So genetic variations, that mean any alteration that can occur in the genetic sequencing among individual, and these can be large, at large size or large scale. For example, aneuploidy, chromosomal abnormalities, or in the numbers, and also can be small as a single nucleotide variant. So the genetic variation, as I said, can be in large scale and in small scale. So large scale as aneuploidies, and then intermediate scales, I would say, for example, structure variants, and in small scale or small size like single nucleotide variant. So what is aneuploidy, or what are aneuploidies? So aneuploidies is alteration or changes in the number of chromosomes. And this is compared, we know that we have a human genome or has 23 pairs of chromosome, or 46 chromosomes in a diploid manner. Okay, so while aneuploidies means that change in the number of chromosome, which can be trisomy, which mean increased number of chromosome, or copy of the number of chromosome, or monosomy, which mean decreased number of chromosome, and this mean loss of chromosome. So, and the aneuploidies can affect not only the autosomal
We are sorry for the break in transmission. So this is a technical glitch from her side. So I'll try to reach out to her again. Allowed to speak. I'll try to, no, presently it's, yeah, we are back. Doctor? Doctor? Sorry for the
Okay, can you hear me?
Yeah, we can hear you now. Thank you.
Okay. Okay, so as I said, these pictures showing examples of normal karyotype in female, for example, compared to the abnormal or aneuploidies or abnormal chromosomal abnormalities in autosomal chromosome, for example, in trisomy 21, which leads to Down syndrome. We know that there is a gain of one chromosome, but while monosomy there is a loss of one copy of the chromosome 20. And usually with the chromosome 20 associated with some diseases and cancer. Okay, so as I say, the aneuploidy also can affect the 6 chromosomes, and there are few diseases related to aneuploidies with 6 chromosomes. But autosomal aneuploidies also show a very severe threat in phenotypes compared to the 6 aneuploidies. So the structure variants include copy number variant and rearrangements. So the structure variant can be described in the changes or alterations in the copy number, in the location or direction of the genetic material. So the direction or orientation of the genetic material. So, for example, and this includes copy number variant deletion and duplication, which include deletion or duplication of more than 50 base pair. And while the change in the orientation of the genetic materials or location of the genetic materials, this includes, for example, the direction of the change in the direction of the orientation of the genetic materials, for example, inversion, and also while the exchange between the genetic materials between two non-homologous chromosomes, this leads to translocation, and both are classified as structure variants, rearrangements. The phenotypic significance or the impact or the phenotypic effects of these structure variants depends on the location of variant and the type of the variant itself. So moving to small variants, which we are going to show the effect, more specifically, the effect of the small variant on the genotype and how they impact the phenotype and they impact the or change the structure of them, for example, the sequence of the amino acid, how some small variant can cause can associate it or can change the a protein sequence, some not the amino acid sequence, and then and some they don't change the amino acid sequence, and how they affect also the protein function and so on. So the small variants would show the association between genotype, phenotype or the relation between genotype, phenotype in causing disease as well. So we know that small variants, and this includes single nucleotide variant, which means a change in single nucleotide. And we know that single nucleotide can be defined as rare mutation, but when this single nucleotide occur in greater than 1% population, defined as a single nucleotide polymorphism. But I'm not going to focus on these things. I'm going to focus here on single nucleotide variant and the impact, so types of single nucleotide variant, the impact on the genotype and the phenotype more than talking about the single, about the single nucleotide polymorphism and so on. So also small variants may include, includes indels, and indels mean insertion and deletion. So changes in the small number of nucleotide range from 1 to 50 base pair. We know that. So the single nucleotide variant can be found in the protein coding region, which include exons, and also can be found on non-coding regions, which they are promoter regions and introns. So the type of single nucleotide variant, and we are going to talk now to explain it now, and also so the type and the impact will be different, depend on where these variants are located. So, for example, the single nucleotide variant in the coding region, so in the exon, will show effect on the protein production. Not all of them, but some of them will show, and we will explain it now. While the non-coding region, while any variants in the non-coding region would affect them, either the RNA expression or the protein level as well, or the RNA stabilities, which subsequently will also affect the protein production. And we will explain that in this picture. So if you focus with me, we understand that this is the structure of the gene, which include the enhancer, promoter, exon and introns, exons. So before starting the protein coding regions, we know there is a 5 untranslated region, and before the end of the protein coding regions, we know that there is a 3 untranslated region. We are going to see here. We are going to explain the effect of different types of single nucleotide variants, but first in the protein coding regions. And I said, as I mentioned before, the protein coding region, which is in the exon of the genes. So as I explain, I put it here in the picture: exon 1, 2, 3, 4, 5, for example. This is an example, and these are the protein coding regions, which mean these regions, or the sequencing in this region will produce a protein. So what is the impact of single nucleotide variant, or different types of different, what is the impact of different single nucleotide variants on the protein coding regions? So I give here an example. Single nucleotide variant detected at the gene coding sequence of exon 3 here. At the 3rd codon. So we know that DNA produce RNA, and then this leads to produce the amino acids. So the single nucleotide variant is detected in the codon number 3. Okay, in the exon number 3. So exon number 3 with this sequence, with this DNA sequence, and if you can see here, one of these sequence are affected. One of these sequence is affected by different single nucleotide variant. In the normal, if you can see here, the codon TGC will give UGC in the RNA, and then will give the amino acid cis, for example. Okay, so if there is any silent variant in the TG, for example, in the TG, for example, the normal is TGC, and the variants become TGT. So affect the T. The change was in the nucleotide, the single nucleotide, or the base T, the base C is changed by or replaced by T. So what is the impact of this change? So the change actually, this change actually lead to the RNA is TG, actually UGU, the RNA. UGU as a codon gives the same amino acids, and that's mean the silent variant change the codon sequence to UGU, but the amino acid sequence is still the same, cis, and this means the silent variants do not affect the protein function, or the amino acid sequence and the protein function as well. And then if we move to the missense variant, with different, actually, as I said, if you compare the normal, the normal sequence was TGC, it changed to TGG. So the G is the, C is replaced by G. So here, this leads to a change in the coding sequence to UGG. And this leads to also a change in the amino acids to become trip instead of cis. And that's mean a change in the amino acid sequence. This change may affect or may not, or may prevent also the protein, may affect or may not. It depends on the location of the change, but may affect the protein function. If we move to the nonsense variant, and you compare, the change was here, TGA, TGA, that's mean the C replaced by A. So what about the amino acid? The amino acid, it changed to UGA. The codon, it changed to UGA. So UGA usually gives a stop codon. And that's mean nonsense variant, it changed the codon sequence to UGA, that lead to introduce cause premature stop codon and lead to termination of the protein production. And this has usually nonsense, it prevents the protein production. So has a significant effect on the protein production. And then when we move to the frame shift variant, so the frame shift variance means insertion of a nucleotide, which changed the reading frame. So during the so for example here insert the T. So this means lead to reorder or the order of the coding sequence. And reordering the coding sequence means that it changed the codings and it changed the sequence of the amino acid. Let us see in the example. So for example, if we go to the TGCT, so this show the example with the frame shift variant, will show you the insertion of the T. So insertion of the T lead to reorder the amino acids sequence. So, and this become, as you can see, change in the amino acid sequence. And this leads to also a change. So this leads to affect the sequence of all downstream amino acid, and lead to, at the end, lead to premature stop codon. So the frame shift has a severe, usually the frame shift variant has a severe effect on the protein function, which we can see here, not only changing the amino acid sequence, but also termination of the protein production. So here, from here we can see, so from these examples, we can see how the single nucleotide variant affects the protein coding region, and how single nucleotide variants on the protein coding region affect the protein production and then also the phenotypes. So the protein production and the function and their function actually in the cells. And this example also a clear show the association between the genotype and the phenotype, and how the variation actually affects the genotype and phenotype. So the scenario may be similar, but in different way. So if it's this single nucleotide variant in the non-coding regions, so what are the non-coding regions here? So non-coding regions means these the one in the regulatory regions, which are regulatory regions, the promoters, also the enhancers, sometimes maybe in the transcription factors, so any single nucleotide variant on the non-coding regions, more specifically in the regulatory regions, can affect the expression of the genes or the RNA level. While also the non-coding regions include the splice regions, which are the introns. So any single nucleotide variant located in the introns could affect the splicing process, and also sometimes affect the protein, not only the splicing process, or sometimes affect the RNA, sometimes affect the protein production. It depends on the nature of, as I said, the type and the nature of the variant itself. So from these examples, we can conclude that the nature of the coding variant and the coding variant and their effects on the level or structure on the function of the protein are essential for variant classification into benign or pathogenic. So that's mean the nature or the type of the variant, where these variants are located, can help us to identify if these variants benign, so they don't show any yes, there is a variant, but they don't show any influence on the protein production, and if they are pathogenic, would show a phenotypic or significant phenotypic effects on the protein production. The production of the impact of non-coding variants, specifically the one in the introns, is still difficult, and that's because these variants located outside the coding regions or the protein coding regions, and also outside the regulatory elements. But I think the sequencing techniques may make things easy to identify these non-coding and non-coding variants, or identify these variants in non-coding regions and their effects as well. So before the end, we can see see how the genotype associated with phenotype, not only for for example, for the or for determining the trait or the phenotypic characteristics like the height, like the high or hair or eye colors, but also in, as I said, in our capability to in increasing or decreasing the risk of developing diseases. Also, we know that not the genotype or the genetic or our genome is not also means the genetic materials of our genome are not only responsible for determining our trait. For example, our height, for example, our weight, all of them actually are also controlled by environmental factors. So that's mean our genetics and and environmental factors, for example, the diet, lifestyles, influence or impact the individual phenotype or individual traits. More specifically, we would say there is Mendelian traits and a complex traits. So some variants, we know that some variants in one gene would cause or would have large size effects to cause a specific disease, for example, sickle cell anemia. While the complex traits, and for example, would say an example, type 2 diabetes. So type 2 diabetes needs many variants in many genes combined with the environmental factors to induce or to produce the phenotype, which is the disease, or the phenotype which is the disease type 2 diabetes. And this is and from this also we can say that the variations in the DNA between so the variation of the genotype, I would say the genotype combined with the environmental factors are responsible in determining our phenotypes, except for the Mendelian trait. So thanks for the multi-omics technology that help us to study the association or relation of genotype to phenotype at different levels, at the DNA level, using sequencing, for example, the DNA sequencing to study genomics, at the RNA level by the transcriptomics using the microarray sequencing, at the protein structure using the mass spectroscopy in that to analyze the proteomics, for example, and also at the metabolomics or at the metabolites and all the chemicals materials that involved in the cells to, for example, to regulate specific function of the protein, using also the high resolution mass spectroscopy techniques in the to analyze the metabolomics data. So I would to say thank you very much for your listening, and if there's any question, we would answer it, I think, after the end, at the end of the session. Thank you very much.
Well, thank you so much. That's exactly almost 1 hour of presentation. We really appreciate the presentation, and this is the stuff of the basic stuff for us to understand as a prospective bioinformatician. So we have to understand the central dogma and also the impact of a genotype-phenotype relationship on health and disease. So I would like to come in from the last part, which is the area of a single change that is having a big phenotypic effect. So, and especially what concerns us mostly in the area of Africa, Middle East, and Southeast Asia. No, Africa, Middle East, and also South America, and also Black Americans too. So it's the area of this sickle cell anemia. It's a scourge that is most debilitating and impactful in sub-Saharan Africa, and especially in Nigeria. So it will be interesting to note that this is just a single variation in the hemoglobin gene that caused this huge phenotypic impact, this phenotypic change. So for those of us who are just joining, so this shows how important the studying these genome-wide association studies, it's also part of that, and also this single nucleotide polymorphism or single nucleotide variation, how effect it has on the phenotypic characterization of any species. So right now, we would want to give chance for questions. I know there will be a lot of questions in the group section. So a lot of people have requested the PPT and I've shared it to the channel. So I want to once again appreciate our our resource person for giving us or taking us through the process. So some are also requesting for the recording. Of course, the recording will be supplied to us after the webinar ends. So now there's a question here, although which is unrelated to what the presenter stated. Well, we would also answer that question. The question goes to us that what is the difference between upstream and downstream analysis? What is the difference between upstream and downstream? So as we await any other questions, so I would like to point out that the presentations will be made majorly on Telegram. So because Telegram we've explored other online platforms before. So but because we felt Telegram is even more better regarding our ability to take in a lot of members, record the sessions, and also share the videos. So it is better and it's free of charge. So we are not incurring any charges. So unlike using other platforms like Zoom, so there is a maximum limit of people you can take in. So, but on Thursday, when we'll be having the session of Ensemble Genome Browser, so that is the only one that will be on Zoom. So the link will be provided to us in due time. So there's another question regarding how do we read transcriptomics and metabolomics data? How do we read transcriptomics and metabolomics data? So, Doctor, can you go through the answers to the questions?
So I can answer the questions downstream and upstream. So the upstream means the genes, at the 5 ends, but downstream at the 3 ends. Okay, so this is the thing. While with the one with the genomic and proteomics and what is transcriptomics?
Transcriptomics and metabolomic data.
Yeah, so I'm not experienced on that. I don't have experience on that, but the thing that I know, genomics, studying them or analyzing the data based on the DNA level, proteomics at the mRNA level, transcriptomics at the RNA level, proteomics at the protein structure. And the metabolomics at so identifying the metabolic markers. So this is the things that I actually know, but I don't know the things in depth, to be honest.
So there's another question. It says that what is the function of the capping and poly A tail in the transcription process of DNA?
Okay. So as I said, so the capping is like a protection from. It's like both are assigned for mature RNA. So this is the thing. So that allow RNA when transfer from from when move from the DNA from the nucleus to the cytoplasm. So these two signs protect the mature messenger RNA from degradation. Okay, so these are the signs that this mature RNA are need to proceed to the translation. Okay.
Okay, thank you so much. Another question states that can you please shed more light on single nucleotide polymorphism?
Okay, so as I said, is it still single nucleotide? Is a change in the single nucleotide? That if we have a population, so if any population, or we can define this single nucleotide variant as a single nucleotide mollifilisms that's related to developing a disease. This single nucleotide variant has to be found in more than 1% of population. So this is the thing that I know about it. Yeah.
Thank you so much. I think that's the question. That's all the questions for now, and the questions has been answered duly. So thank you so much, Doctor, for taking out of your time and expertise to share with us this important aspect of bioinformatics. So, and this has formed an important basis for us to proceed on to the next session. And as it's customary, it will be customary in our sessions. There's going to be a 10 minutes to 15 minutes break in between presentations. So we can have, I know by now it's going to be around 12 p.m. or 1 p.m., irrespective of where we are coming from or where we are joining the class from. So we'll be having 15 minutes break to rest and take our lunch, and then we'll come back to the Anton session, so which will be anchored by Mr. Adebayo Pabumi. So on multiple sequence alignments, the approach and how to go about the sequence alignment. What is the importance of sequence alignment to our bioinformatics analysis? So here we'll be talking about bringing different sequences from different individuals or different animals or different plants or different species, and then comparing or bringing them together using computer tools to align them and then to make a sense out of the combination. So how do you go about that? What tools do you use, and do you identify if you correctly aligned your sequences or you have not? So that is what will be taking us through in the next session. So in the next 15 minutes, that will be 8:30 from my side. So we'll be starting the third session. So the other questions, I'm going to be reserving them. The questions that have not been answered, it will be answered in due course. Thank you so much. So we'll be back in the next 14 minutes. So see you in the third session. Thank you. Hello everyone. So we can start preparing for the next session. It's about to start. Mugweza, I can hear you, sir. So let's as we await others to rejoin back the session, so we can start the next session. So by in 2 minutes time, we'll start the third session, which is going to be a hands-on session, and we are going to be taking through the multiple sequence alignment. So this next session, it's more of a hands-on and not more of presentation. It's going to be how we are going to be shown how to do this analysis, and then not just the explanation regarding the analysis itself. So let's see. So we have, yes, we are up to the number. So the presentation will start in a bit. So I'm trying to get some things done here. So 1 second, please. Please hold on for me. Now, so the session will begin. Now you can start, sir.
Hi, everyone.
Yes, I can hear you, but please hold on, please. I'm trying to. I'm coming. Sorry, we are trying to sort out this audio here.
Right now, through the sequence alignment methodologies and approach, and as equally said, it's much more of a kind of hands-on, and not to be too fast, so that it can be able to carry everyone along. But before I start, let me say a big thank you to the Noblekinmat Bioinformatics group for kindly wanting to share my little knowledge with this great audience on this platform this day. I'm grateful and grateful. So let's begin. Sequence alignment methodologies and approach. Sequence alignment methodologies and approach. I've not really been following the workshop because of some other equipment, but I think by now you should understand what sequences are. I think by now you should understand what sequence or I will not waste much of our time with demonstrating what is the multiple sequence alignment now. Multiple sequence alignment. Alignment can actually be occupied. We have the right. We have the sequence alignment.
Can you hold on a minute?
We wouldn't be holding ourselves with the pairwise. We only be doing the actual sequence alignment.
So, please, can you hold on a minute, please? I think there is a lot of feedback. It's kind of echoing again. How do we go about it? Can you please remove the earpiece, probably? I think I will try. Maybe you can try that aspect.
Okay, what about now?
The feedback is no more there.
Okay, it's better like this.
It was better than the initial one.
Okay, so can we try it again?
So I think what you should do is that you can use an earpiece for the phone.
Okay. You mean I should remove the earpiece for the phone?
I think the feedback is. Oh, sorry. For guys, I'm sorry. The problem we had was that the audio from resource person is not that. It's kind of mixed up. So we are trying to connect the audio together. So please give us some few seconds to sort it out. So try to you can put the earpiece in the system and let us see if it's going to work. Okay, you can try to speak now.
Okay, so Doc, can you hear me?
It is now better. You can continue now. I think the sound is better now. It's better than before. Okay, you can go on now. You can speak now.
Okay, can you hear me?
Yes, I can hear you now.
Okay, and I hope it's better now, right?
It's much more better than the initial one. So if you can hear that clearly, kindly comment so that we can proceed. Yeah, it's good now.
That's right.
Yeah, you're good to go.
Okay, so as I was saying, sequence alignment can be said to be of two types. We have the pairwise sequence alignment, and we also have the multiple sequence alignment. But in this training, we actually be doing the multiple sequence alignment. Because once you're able to do the multiple sequence alignment, definitely you'll be able to do the pairwise sequence alignment. So let's get to our browser. Please get over to your browser. Can you see my screen?
Yes.
Good. So let's get over to our browser and let's quickly download the tools we'll be using for this hands-on. Actually, there are a series of alignment tools, but for this training, we'll be making use of the Bioedit. We have Mega, we have Codon, Code Aligner, we have Maft, we have Cluster, Omega, etc. So much more of these alignment tools. But for the sake of this training, we'll be making use of Bioedit. So for some of us who don't have this Bioedit already downloaded and stuff, let's quickly do that. Just go to Google. Good. Then you type Bioedit. And you click. Then, as you can see here right now, you can see Bioedit 7.2 download. Actually, it is free. This tool is free for download. So you click. And as you can see, towards the right side, you can see download. Then you download. Because of the fact that I already have the software installed on my system, I wouldn't be installing it. But as you can see here, it's already downloading. So when it gets downloaded on your system, just try to click and install accordingly. So I'll be pausing here for a while so that we can be able to quickly install the software that we have downloaded, the BioEdit. So when you are done with this, from the chat box, so that the admin can actually bring every one of us together. So the downloaded BioEdit, kindly follow the instructions given accordingly and install on your system. So when you are done with that, please kindly respond on the chat box and let us know that you are done with that so that we can proceed. I hope you are all getting it.
Yes, we are.
Good. The moment we are done with the download and the installation, please let's keep your so that we can proceed. Once we have like 5 to 8 responses of downloading and installation, then I think we can proceed. Please, we are waiting for your responses.
So we have one download complete already.
Good.
So
Can we have more responses, please?
another response. We have the second complete download.
So don't be afraid. Just take it as follow the given instructions. You go to Google, you type BioEdit, then you click, then you download, and lastly you install. Following the appropriate instructions that will be coming up on the screen. Very easy to install, and I hope by now we should have considerable numbers of individuals who have been able to install the BioEdit.
Yeah, I think we can proceed now. Others can join.
Can we proceed, please?
Yes.
Can we proceed?
Yes, we can.
Okay, good. So for those of us who have been able to install, just click the BioEdit and open the software. You will see something like what is displayed on the screen now. You will see something like this as displayed on the screen.
We only have your Google Chrome page displayed, sir.
Okay, So let's go back to Google. And now, since we have been able to install the alignment tool we're making use of, then let's go back to Google to download our DNA sequences. For the sake of time, I would have said you guys should be giving me options of organisms that you would have loved me to download their sequences, but notwithstanding, just follow me accordingly. Now, this morning, or rather, this afternoon, or whatever it is, in your area, we've been making use of 6 DNA sequences from 6 different organisms. And of course, we've been making use of the mitochondrial cytochrome oxidase subunit 1 gene sequences. We've been making use of these mitochondria CO1 gene sequences. So we've been starting with that of human. So just type NCBI. That is the database, from which you'll be downloading our DNA sequences from. I'm sure that by now you'd have been accustomed to what NCBI is. As you can see on the screen, it's loading. Now, when you get to this, do we all have this on our screen now? Now, you can see here that you can see all databases. So just click and then come to nucleotide. Just scroll down after clicking and click nucleotide, because we're actually working with nucleotide sequences. So you click nucleotide, and after you might have done that, you type in the search box here. You can see search. So you type Homo sapiens CO1 and you enter. Or rather, kindly put mitochondria, of course. Mitochondria. Okay, let me do that again. Homo sapiens mitochondria CO1. Okay, it's saying
Yeah, we're still here. So I think the network has an issue from our presenter's end. Oh my God. So so that we won't take much of our time. So I would continue from where the presenter stopped. So I'm going to share my screen right now. So our presenter apologizes a lot for the break in transmission. Okay, I think we can all see my screen right now, right? So if you can see my screen, please notify me. If you can see my screen, I'm on my Google Play, Google Chrome screen. Please notify me if you can see my screen. If you can see my screen, please notify so that I can proceed. Don't worry, I will take it from the beginning again. I want to share my screen, so please notify me if you can see my screen. Okay, thank you so much. I will do that again. I will start from the beginning again. So now I'm sharing my screen. I'm sharing my Google Chrome screen. So now what our presenter did was that we downloaded the software BioEdit. So BioEdit software, we search for that on Google Chrome or any browser we are using. So we'll click on the Google Chrome and then on the link, and then we'll download the software. Then after doing that, we'll move on to the next point, which is we are going to search for NCBI. So NCBI, okay, let me go the easy way, the preliminary way. NCBI. So NCBI is a platform whereby all this information data is a database for sequences, for a wide variety of bioinformatics analysis. We will be basically focused on NCBI. So you will need this tool. If you are new to this aspect of bioinformatics, know this name, NCBI. So the full meaning of NCBI is National Center for Biotechnology Information. So, and you click on this. So the website, I'm going to provide the website on the link on the group. So you can click on that link and then join. You can use that to join the to log into. So this is the NCBI website. So click on this, and then what we want to do is that we want to focus on the CO1. I can see some comments coming in on the fact that they are using Mac OS so system. So actually, I think there is an option to download for OS download. Yeah. So if you scroll down, there is BioEdit for Mac. So operating system. So there is a, if you click on this, because I'm not using Mac, so I'm still a poor student. So so if you want to use for Mac, so OS, you can click on this BioEdit for Mac, and then the appropriate link for you to download will come up. So let us assume that we have all downloaded BioEdit. So then we'll move on to the next part, which is coming to this wonderful website, which is the NCBI website. I think we are now clear regarding the OS and the other tools. So for those who are using Linux, I'm not sure if you are a beginner, it's just my assumption maybe. So you might not, as a bioinformatician. Okay, I know the questions might come, which is the best system to use for as a bioinformatician. So one of the best system to use is Linux system. It's not even one of the best. So in ranks of what you can use, the tools, that you'll be able to use by the operating system with which you can use for bioinformatics analysis. So most advanced level analysis are done with Linux system. So followed by Mac and followed by Windows. However, if you are using Windows, that is not a SATL for you. There is a virtual machine software that you can use to perform Linux or Mac operating system that bioinformatics analysis that requires operating system on by Windows. So that is why that is the rank of things. So if you are using Windows, you have no problem. If you are using Linux, good for you. If you are using Mac, wonderful. So everyone is served in this area of bioinformatics. So yes, so for Android, you cannot perform bioinformatics analysis on Android. I don't know the kind of Android you are using, but I have not seen it so far that you'll be using Android for bioinformatics analysis. So let us move on to searching for the NCBI. For the NCBI. So I think presenter, okay. So let us move on to presenter is deeply apologizing. It was a technical fault from the system. So we are very, very sorry for that. So we are very, very, very sorry for that. So now we'll move on to the next point, which is searching for the sequences that we are going to use for our analysis. Just like it was introduced to us, what we'll be doing is multiple sequence analysis. Now, some people are new to what a sequence is. Now, what we want to do is that we want to download sequences from NCBI. These sequences have already been submitted to NCBI by researchers who extracted DNA and sequenced the DNA. And then these sequences are deposited on this website, on this database for analysis. Now, I would search for, okay, click on nucleotide, just like we've been told to by the presenter. Okay, let us leave it as all database. So we can search for Homo sapiens CO1. So let us search. If you can't hear me, please notify so that I can adjust. If you can't hear me, please notify. So now the result for searching for Homo sapiens is out. Now we are going to be working with these Homo sapiens sequences of CO1. For those who don't know, or for those who are new to biological terms, so cytochrome oxidase 1, a quick search of cytochrome oxidase 1 is a gene that is present in the mitochondria. And the mitochondria is the powerhouse of any cell, any animal cell. The equivalent of that in plants is the cytoplasm, is the chloroplast. So in the mitochondria, which is the powerhouse, there are 13 genes. 13 genes. Don't worry about the terminologies if you are new to biology. There are 13 genes, but one of the most important genes is cytochrome oxidase 1. So this cytochrome oxidase 1 is used popularly in population studies. It is used popularly in population studies. So that is why we are searching for cytochrome oxidase 1. And why do we want to search for cytochrome oxidase 1? We want to carry out multiple sequence alignment with these sequences. Now, the reason why we added Homo sapiens is that the CO1, when we search for the results, when you search, the result that is going to be displayed is going to be that of human beings, Homo sapiens, human beings. So the CO1 for human beings. If I put another species here, the result would also come out as another species. So that is why we are having CO1 for Homo sapiens here. So, and then we have the results. So these are the results. There are different results, many results. But which one is which? Which one are we going to work with? So we are going to be working with the ones of nucleotides, just like we've been told to search for initially. So we are going to work with nucleotides. Are we following? So please indicate in the chat box if you are following, so that I can know that I'm not presenting to myself. Please indicate. Thank you. So I'm not talking to myself after all. Thank you so much. So thank you so much. Now I'm happy. Now we are in the nucleotide part. So this nucleotide, invariably, what we have here is that when you search this keyword, even if you are in anywhere in the world, if you come to this website and you search this keyword, the result that you are going to have is that there are 2,777 nucleotide sequences of CO1 in NCBI. So we are going to click on this. When you click on this nucleotide, so it is going to take us to the page whereby we are going to be working with what we need. So we are going to be working not with everything, not with 2,777. So not with 2,777, we are not going to be using everything. So we are going to be using just, let's say, 40. As a beginner, if you are just a beginner coming up or starting out with this type of analysis, what I would advise you to do is that you should start small. You should start small. Don't work with too much data set. So start small. Now, what we are going to be doing here is that you can see that these are the results. Homo sapiens cytochrome, blah, 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 blah. Here we have Anopheles argentis cytochrome. So now you can now sieve the data that you need and remove the data you don't need. We don't need Anopheles argentis. We are looking for Anopheles. We are looking for Homo sapiens. So Homo sapiens. So we have different Homo sapiens sequences. So yes, Homo sapiens sequences, they are different. So, and then another thing that you are going to watch out for is that you can check for the length of the sequences. Dr. Aisha has already explained to us that DNA sequences are represented by A's and G's and T's and C's. So these are the 4-letter words that makes up a sequence, a DNA sequence. So the combination of these 4-letter words is what makes the length of a sequence. So now when we want to select different sequences, so you can click on this box, 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 click on this box. So if we keep on clicking, it will take us so much time. But the problem is that we have in this page we have 20, only one that is odd one out. Just select, just come to this part whereby you select. So let me unselect everything so that we can see. Now we want to download these 20 sequences. We know that there is an odd one out and we don't need it. So we can just come here, click on send to, and click on file, click on this part where they have summary and click on faster. I will take it again. Now I'll come back for those who are just to break it down. So we've downloaded this software. So this software is called BioEdit. It's one of the tools that is used for multiple sequence alignment. So if you are using Mac, you can download for Mac. I'm not sure there is another one for Linux, but I think there should be. If we search, we can see. Let me search bioedit for Linux. So bioedit for Linux. Oh, there is no bioedit for Linux, I'm afraid. I'm afraid there is no bioedit for Linux. Well, anyways, I'm still going to teach us another method for uh for multiple sequence analysis, so that doesn't require you downloading any software. So now oh, you can, oh, some people are mistaken by writing CO1 or COI. Actually, it is same thing. See, you can, but in this case I wrote COI, and if you write CO1, you are going to get a different result, like a different result from what we get if you type COI. So that is why it is very important you know what you are searching for. So we came to NCBI, and then we are here. We are searching for this particular result, and we got the result. So we clicked on the results, and then we are going to download all these 20 sequences, or only 20 sequences. So let us try to download. You come up, you scroll up, you click on send to, leave it as it is, complete record, yes. Now click on file, don't say because it has been clicked already, click on file. If you click on it, there will be a drop down. Now it would ask you download 2777 items. No, we are not downloading that. So now you come back here, you click on the ones that you want to download. So we want to download only this, so you click on that and you select the ones you want to download. So I'm going to be working with 20 or 19 sequences so that we can start from there and gradually. So for more, okay, I'm going to explain that later. So this is for the full download. Now you clicked on what you want to download. Now come back to the send to, click on it, complete record, file, click on file, and then you can see that the options for you to download has changed. It says download 19 items. So, and we've selected faster. It is important. Click on faster, click on faster, click on faster. So fast A files, some people call it fast A files or faster. I call it faster. Fast A file is too much, it's too long, so just call it faster. So now it says okay, this is another functionality that frankly speaking I've not explored. So it says that default order. This default order means that this one after this one. So this one says accession number. So this one will arrange according to the accession number. Should we do that? You can do that on your own. I'm still going to try this. I think this is going to be useful for me. Now, also this one says download according to sequence length. So no, okay. Should we? Yes, no. Well, let us click on sequence length. I've not tried it before, but don't worry, it's not a problem. And I'll tell you the advantage. The advantage is that you would notice that our sequences are of different lengths. We have 386, 457, 334, 637, 636, and so on and so forth. So we can click on sequence length. It will arrange it according to the sequence length. So now we can create file. As soon as you create file, there will be a download, but it will download your sequences. Now let me reshare my screen. Okay, you can see my screen. So, but I'm going to share all my screen so that you can have everything. You can have everything. So here I am. I'm going to share all my screen. So now the download is already made. Now when I click on this, I open this file. So as you can see, the sequences are arranged in according to their sizes. If you can see this notepad, please let me know so that I can be sure that my screen has been shared successfully. Kindly indicate if you can see the notepad. Please indicate if you can see my screen notepad where these sequences are displayed. Thank you so much. I have a confirmation already. So that means I can proceed gracefully. Thank you so much. Thank you so much. Okay. Now we'll proceed to the next stage. And what is the next stage? Now we have all our sequences downloaded, and then we can proceed. So how do we proceed? We've saved our sequences. Okay, I should take it again from the selection. So, okay, let me back it a little bit. So we came to NCBI, we searched this particular keyword, and then we got this result. These are the results. So I came to nucleotide, I selected nucleotide. Okay, I selected nucleotide, and then these are the results. It gave me 2777, but I don't need to download all the 2777. I don't need them. So I want to select. So if I want to download all the 2777, I will just come to send to, click on complete record, click on file, and then it will tell me download 2777 file. No, I don't need to download that. I opt out. So I will now click on the box here for the sequences I want to download. I will just select them one after the other. I'll select, I'll select, I'll select, I'll select, I'll select, I'll select this. I want to download 20 as a beginner. I don't want to make the work cumbersome for me. I'll select this, and then I'll come up again. You can do it down. You can select from here, from top or bottom, from top or bottom, top or bottom. So come here, complete record, click on file. Now what we want to download is the FASTA file or FASTA file. So I will leave it at default order. You can select any order irrespective of whatever you want to do. Just don't worry yourself. It's nothing. That's not our concern. So default order. So, and then I click on create file. If I click on create file, the download will come. So now I have the notepad displayed here. So that means I have all my sequences here. I can play with them as I wish and as much as I want. So yes, the reason why is that you are not searching the right keyword that we searched for. So that's why searching for the right keyword is very, very important. So for the person asking that he or she got 0 result from the NCBI search, so the right keyword matters. So now we move on to the next level. I think we can now easily move on to the next level of the analysis. Only one was not selected out of the 20 sequences that was displayed. Only one was not selected. Only one was not selected. So we'll move on to the next stage. So we'll move on to the next stage. So please, if you are just joining, you can revisit so that we would not waste much of our time. Thank you. Now open your BioEdit. So when you open your BioEdit, mine is already opened. Mine is already opened. Have we opened ours? If you cannot open your sequence data, that means you clicked the wrong file type to download. You must okay, if you still cannot open faster through Notepad, some do not, because you are starting for the first time, right click on the download data. If you right click on it, go to open with, it will bring different option of software to open your sequences with. So open it with Notepad, although it can also open with Microsoft Word. So open it with Notepad. So I think we can now proceed. Now come to BioEdit, click on File, and then click on Open. I'll take that again. Click on File, click on Open. I'll take that again. Click on File, click on Open. And then go to the file where you downloaded your sequences to. So come to Downloads. Mine is in Downloads. And then I can select the sequences. I hope. So you can see your sequences here. Now, since I downloaded according to accession number, it will arrange my sequences according to accession number. So if you can see, this is from MH791068, 067, 066, 065, 064, 063, 066. So most likely these sequences are from the same source, are from the same individual that submitted them to NCBI. So others are different, only these sections are the same. So now we have these 19 sequences. You can see the number of sequences, 19, total sequences 19. So the remaining work for you to do is not much any longer. You don't have much work to do again. Why? You just come here to accessory application, come to accessory application, click on cluster W, and then you click on run cluster W. I'll take it again. Accessory application, cluster W, run cluster W. I will take it again. Accessory application, cluster W, multiple alignment, and then you click on run cluster W alignment, click on OK, and then you wait for the system to run. So if you did what I did, if you do what I did, yeah, that's the right grammar for that. If you do what I did, you get a result similar to this. You can see what we mean. Now, let me take you back a little bit. Initially, when we match all our sequences together, this is how everything looks like. It doesn't have, like, you cannot say any information with these sequences because they are different. but from the same species. It is from the same Homo sapiens, but they are different. You cannot tell any information from this arrangement. So what this system does is that it now aligns the sequences with each other. It aligns them with each other. It brings them closer to where regions where they are similar. All the regions where they are similar, it brings it closer. It brings it closer to each other, so that you can now infer your biological analysis. Now, and this was the result of that. So, and this was the alignment. So basically, this is the end of multiple sequence alignment. Basically, this is the end of multiple sequence alignment. So what this tells you is that all these sequences, you can see that they have regions whereby all of them, they are the same. This is one, and you can see that they are also the same in this region too, except one prodigal son, one prodigal sequences. So the next step is for us to trim our sequences and remove the faulty sequences that will affect our analysis. Now, remember that I said initially that some of our sequences are longer than each other. So some are 300 and something base pair long, some are 1000 base pair long. So here you'll be having problem because the location whereby they are similar is a little bit different from one another. So you have to look for a middle point whereby all of them look are similar, and then you can edit them accordingly. But for a start, we can work with the fact that we are going to remove the faulty sequence. So how do you remove faulty sequence? You come here. Let me enlarge this part. Let me click outside. So you can see the faulty sequence out of all these sequences. The most faulty one that has fault, you can see it is having breaks in between. So now a question came that what is the size of the sequence document? Now the sequences, the size varies from one each other because they are not sequenced from the same person. Even if it is from the same person, there will still be slight differences, very very minute, maybe, but basically it will be same from the same individual. You would have no problem with that. But now we are comparing from the same species, which is the human being, cytochrome oxidase. So the sequences are different, the lengths are different because one, the sequence quality, two, yes, basically the sequence quality, and then the quality, the sequence quality, and the quality of the DNA that was sequenced. So that's the difference that makes the length different to one another. So we are going to remove the problematic one. So you click on this part, which is the problematic one, you right click, and then you delete sequence. So once you delete this sequence, you come again and check if there are any faulty sequences too. So ladies and gentlemen, what we are doing now is that we are editing our sequences. We are editing manual, we call it manual trimming or manual editing. Now I've also seen another faulty sequence, and that faulty sequence is sequence number 2 on line number 2 and sequence number 1 too. Sequence number 1 and 2, because they are not as long as the other ones, and what we are not looking for, we are just doing as a free roll, so we are not particular about anything yet. So you can select. How do you select? You can click on this and drag. You can see the way I clicked and drag. I clicked on the first one and I drag to the second one. You can try. It doesn't, you can't, if you are having problem doing it at once, you don't worry, it would not take long before you get used to it. So you click and drag, click and drag, you can select. So we selected the first two sequences. So And then what do we do with them? We right click and then we'll delete sequences. Now we are looking for 40 sequences because that one will affect what we want to do. So we have another 40 sequence. Okay, for the person asking 40 sequence, so I will show how to delete again. So let us delete these ones that are short. So you click, drag, click, drag, click, drag. If you want to unselect, you can click outside that. You can click and drag, and then right click. You right click on that sequence and then delete sequences. So that is how to do it. Now, a question asks that can you check the quality of the sequence before downloading? Yes, but that is when you are dealing with small number of sequences. If you want to work with about 100 sequences, you might not have time to check them before downloading. So just download and then come here to manually edit. So the reason why BioEdit is very useful is because you can manually edit and trim whatever you want to use, whatever you want to do. So that is why BioEdit is very important. Now, let us move on to the last part of the multiple sequence alignment. It doesn't take long. Now we are moving to the last part. Now we want to trim. We want to trim our sequences. Now, how do you trim? You can see that the outer end of this part is very jagged. Like they are not the same. They are not uniform. And the outer end is also not uniform. So what we want to do is that we want to trim and then make all of them look nice and good. So you can come here, come to the beginning part. Before you do that, click on mode. On the mode, click on mode and click on edit. Once you do that, you'll be able to remove whatever you want to remove from this part. So click on mode and click on edit. Click on mode and click on edit. Click on mode and click on edit, 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 edit. Now we have done that. Now we want to remove and we want to manually trim. So what I did was very simple. I place my cursor close to the top here, close to the top here. I click and then I drag. I will do that again. I click on this and then I drag. Now, where are we dragging into? Now we are dragging into the point. We are dragging into the point where all of them are the same, and this is it. From the top to the bottom, all of them, this is where the sequences started becoming uniform. This is the point where all of them started becoming uniform. So now this is the point we are going to stop. The last one before the uniform part. So if you want to edit again, you click on mode, you click on edit, click on mode, click on edit. Once you do that, you are good to go, and then you come here. Now we've identified where our dragging is going to stop from the start. So you're going to do it gradually. Once you selected, let me take it again. So once you drag and you reach this point, all you need to do is just press backspace. Backspace will remove everything. So, and then you come here and then you stop here. So this is the point whereby they stopped being different, and then you press backspace too. So the forward part is done. These are spaces. You can remove the spaces too. Backspace. You can select the spaces too, and then backspace. So you can see what we are trying to achieve. We are trying to achieve a relative number of uniformity. Now, in this part, this is the back part. So you are going to see that this is the point whereby all of them stopped being uniform. So you are going to drag also and then delete, drag and 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 delete, and then we are good to go. So this is beautiful. This is beautiful. If you got to this point, congratulations, you are now a successful uh, multiple sequence aligner. Multiple sequence aligner. So now, what I did is not magic. What I did is very, very simple. I tried to check the differences between my sequences. I tried to check the little differences between my sequences. Dr. Aisha talked about single nucleotide polymorphism. So single nucleotide variation. This is an example of nucleotide variation. All of them, they are the same, except in this point, all of them, they are C, 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 C. This guy had A. In this point, all of them, they are C, 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 C. This one had T. Here, all of them, they are T, T, T. This one had C. So that is an example of single nucleotide variation. So we can check them in our sequence. How I did that was that I came here. This was the initial page. I came here to this particular pane, to this particular panel, and then I selected this TTTTT dot 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 dot. So this would show it this way, and then this is the end of multiple sequence alignment using bioedit and using Homo sapiens. Ladies and gentlemen, do we have any question? Do we have questions? Now, to conclude on the multiple sequence alignment, you have to save your file. How do you save your file? We come here to File, we click on Save As, and then you can name it any name. Noblekinmat fourth cohort multiple sequence alignment. So we can save. So I'm going to take it all over again for those who are yet to understand. So I would assume that we have been able to download our web sequences, and then we've been able to download our software. So this is File. Click on Open. So click on Open. We've downloaded our sequence. Click on it. Open. And then these are the sequences. So they look jagged. They look jagged. So you should be able to see Save As. When you click on File, you will see these different options. You will see Save As. Save As. You should be able to see it, Save As. So now we are here. I want to assume that I'm taking it from the recap. So what we want to do is multiple sequence alignment. All you need to do is just click, 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 and that's all. So you click on accessory application, cluster W, run. I'll take it again, accessory application, cluster W, run. I'll take it again, accessory application, cluster W, and then run. So click on OK. In a few seconds, your results will be out. So the result is here already. So Now we'll move on to the next stage is editing our sequences. So you can see the multiple sequence alignment. We'll first of all check the sequences that will give us problem, faulty. What do we mean by faulty? So the sequences that do not look like the others, yes, they might be an outlier, but most times, in this case, they are not outlier. That means the quality of the DNA is not that good, so it will affect what we want to do. So we'll click on this, you right click and then delete. We are identifying the sequences that are faulty. So we've also identified the sequences number 1 and 2, which are very short compared to the others. We'll click and drag, we'll right click, and then we'll delete. And then we'll look for another one that is we are considering the length of the sequences now. So we click on this also. We've already seen some sequences that are faulty. So this particular tool, click and drag, and then we delete. And then we'll come down back to, is there any other one? No, everything is good and fine. So now we'll move on to the third stage, which is the manual trimming. And how do we do that? You come to mode and select, you click on edit, and then you click on here, and then you drag to the point they started being the same. So you can do this and you can see that clearly. If you click on backspace, you can remove these spaces, but sometimes it's not needed, but it's good to make your work have a good finishing touch. And then now you come back to the back, so you can see that this is where your sequences started being different. So you can remove, you can remove, you can remove. These sequences are too long. You can remove, you can remove, you can remove, and you can remove. So that's it. So we're finished the manual trimming, and then we want to save. So when you click on file, you'll see different options: new alignment, open, new from clipboard, mute as text, save open, save as, blah blah blah. So you click on save as, and then you can type the name, any name you want. So you can type, leave it as faster, don't change it as faster. So don't change it as faster. So and then you can save. So then that is the end of multiple sequence alignment. So for those asking for the video, so I will share the link to download the video at the end of the class. So I think we have 14 more minutes till the end of the class. So if we have more questions, so regarding the what's it called? So regarding the recordings, so I'm going to share it, and then I'm going to share it to the group, and then we can have the recordings. So do we have any question? So I think I've seen some questions in the group chat, so but I might not be able to answer them right at the moment. So questions. So for bioedit, there is no score for matching nucleotides. When you create a file, then you will have your sequence. Yes. When you create a file, the sequence will download automatically. There is no need for save as on from the NCBI. Once you create file, everything will download. For Bioedit, click on File and Save As. So just change. All you need to do is just change the name or save it to the name that you are using it with. Yes, Notepad is basically a text editing tool. So don't worry. Maybe because you've not been using it before, that's why you had problems doing that or using it. I know by now you'd have gotten used to it. So are there any tools for aligning sequences? Yes, there are different tools for aligning sequences. So unfortunately, we would not be able to go through them presently, but I am going to share the link to our previous sessions that we discussed that. So another question talks about, okay. The app does not work on Android. If you want to compare similarity between many sequences, there are other tools you can use, but you have to pass through the Bioedit first to make it easy. So I think we discussed that too in the last sessions we had. But yes, I will share the link. So how can you know if the sequences are faulty? Yes, you can know depending on what you want to work with. You can know what you want to work with. So how you know is that, for example, in analysis presently, we are particulate about the length of the sequences. So we don't want short sequence, we want long sequence. So had it been our sequences are the ones that are short, we would have no option than to compare our sequences with we compare our sequences with the ones, and then we will trim accordingly. So don't make any changes to the processes. You only have to click on, just click like I've showed you. So you don't need to change anything. So in the absence of no questions, I would like to draw the curtain for today's event. God willing, we are going to meet tomorrow, and please kindly I'll send the link to download tomorrow's software tool, which is the site. Okay, tomorrow's okay. We are not downloading any software tomorrow, so don't worry. So let us come to early, just like we did today. And thank you so much for being a part of the program for the last 4 hours. So I really appreciate being here. So 3 hours 40 minutes precisely. So thank you so much. And if you have any more questions, you can try to reach out to me personally, but I might not be able to answer directly after the class. So, but drop the questions on the group. The people that are capable, if I'm not able to answer, they will be able to answer your questions. So if I'm trying to copy, so no, it won't affect your results because we are still going to get to that. I'm going to share the link to the past class that discussed in details about trimming, manual editing, and the likes. So yeah, home and drive. So thank you so much. We are going to end the class now. I'm going to stop the recording. So officially the class would end now. So thank you so much for being a part. It's been a pleasure from the beginning of the class. Thank you so much and see you tomorrow.